Yeah.
Yep.
Great. Okay.
Thank you, Valerie, for that introduction. It could have been way more embarrassing. I had no idea where you were going with that one. I'm Laura Adang. I'm a child neurologist here at CHOP and new to the 4-H community, so thank you for being so embracing of me and the things that I think maybe we can bring from my experience with other leukodystrophies into this space. And today I'm going to talk a lot about hope. Dr. Wolf did a beautiful overview of the work that she has built over the last decade plus that really underscored a lot of the gaps. And I'm going to take a different disorder that we have worked on for around 6 years now and show how we have identified the gaps, very quickly mobilized a team and network of collaborations between families and physicians, and really moved this ultra rare disease into a space where we are developing therapies. And that's where I really hope and anticipate we're going to be as a community in the coming years. I do have a couple conflicts of interest in rare diseases. While patients are rare, doctors that work on it can be oftentimes even more rare, so we many times have disclosures to do. I do work with drug companies and have various NIH fundings, and I work with several different rare disease organizations for funding as well. I wanted, because I'm new to the community, to share just a moment about me and who I am. I'm an MD PhD. I started off in Georgia, so if my southern accent comes back, please forgive me or congratulate me. I was a biochemistry major, and then I went to UVA for my MD PhD, and I thought, oh, I'm definitely going to do adult infectious diseases, because when I was little, it was when the Ebola virus was coming out, and I was like, I'm going to be a virus hunter. Little too topical now, but I really wanted to do that. So I did straight infectious disease. I have a PhD in virology and looked at how viruses engage with the immune system. And I accidentally fell in love with child neurology and then fell deeper and deeper in love with the community of rare diseases. And I thought, okay, well, maybe from my basic science PhD, I learned how to love science, but I won't come back again. But I actually work on biochemical disorders that affect brain development, and I develop and work with people to develop viral vectors. And I also work at several immune dysregulation genetic disorders that mimic congenital infections, including herpes viruses, which is what my PhD was on. So it shockingly has been a very linear journey and what felt like wasn't linear at all. Today we're going to talk about that hope that is building in our community. As I'm telling a room of people that know this even better than me, 5 years ago, 10 years ago, there wasn't as much movement. And now clinical trials for these rare diseases are coming so fast, I have to check before I walk into every single room to make sure there's not a new clinical trial that's out there. And so where it was a space of very little development, we now have tons of both drug repurposing strategies, gene therapies that are being developed, ASO technology. It is a place of incredible scientific growth. And today we're going to talk about 4-H, but more than a dozen leukodystrophies now have treatments that are in development and really very, very far along in development. When we used to think about leukodystrophies, very few of them had known clinical courses, had treatments. And now, from this example of these early leukodystrophies that were among the first to be described, we now have gene therapy that's approved, enzyme replacement that's in trial, gene therapies that are in trial, ASOs that are in trial. Again, building hope. This is a place where, while we are doing this first in our community, we're in a larger community that has helped to pave this way for us. And so we get to learn from the other people and the mistakes that have been made to hopefully have a less bumpy journey ourselves. One of the distressing things as a leukodystrophy doctor is when I joined, there were 30 approved different leukodystrophies, and I'm like, I got this. I can memorize 30. That list is getting bigger and bigger. And so while there is a lot of hope, there's also a lot of diagnostic improvements, as we talked about today. We are beginning to understand, as we're sending bigger and bigger tests on people, we're both recognizing new leukodystrophies and recognizing a spectrum, which we call phenotypic expansion, that we had no idea existed. But spoiler alert, we know it exists. This happens for every single disorder, right? This is what happens. You start testing more, And then you identify people that are affected in different ways. And our job is not to narrow our view of what a disease is or what a change is. It's to be able to predict the subtype very early. And for me, that helps to give better guidance to families, but helps me to design clinical trials. The ones that are shown in blue all have clinical trials that are active and ongoing. So this is, again, a place where understanding that phenotype, meaning how it affects people, and also particularly as that is growing and changing over time, it's essential. Because we can take a dish of cells and we can start screening therapies through high throughput, and we can develop mouse models, and there are 2 great mouse models for this disorder. And then we can try and take it into clinic. But the challenge is if I don't know who to ask the question on at the perfect time, we could have a therapy that works, but our question was not a good question, right? It is just as important as developing the gene therapy is understanding, okay, I need children between the ages of 4 and 6 with this specific change, and I'm going to measure it in this way. And that's how you get a positive study, right? Because you have to understand what's going to happen to the best of your ability. And so what I do is I work on phenotyping and natural history studies to help to pull apart what seems too complex and make it as simple as we possibly can. We do biomarker work and validation, and there's ongoing work in 4-H. This helps us to know that we're measuring the right thing. Sometimes biomarkers can help us predict the future, at least be something that adds in. And importantly, they also can sometimes let us know that a therapy or a treatment has gotten to where it's supposed to get to. And so as we move into clinical trials, we need all of these arms, and we're working on it, right? This is an area of growth that goes along with the beautiful gene therapy work that's happening. And then I want to circle back. Another reason I really, really want a biomarker is I want newborn screening someday, right? And that is really going to be how we change the face of what it means to have 4-H, is when we can identify children early and offer them a treatment that's transformative. And those are the goals of where I'm going to set the bar for the future, right? I want earlier diagnosis, and I want treatments as soon as I know what I'm helping families with. But there are challenges. It's rare, sometimes ultra rare. Sometimes we don't even know how rare it is. And so we also get this phenotypic expansion. What we thought really defined it is much bigger than it is. It always is this way, right? So don't feel disheartened that it's much more heterogeneous or there's this bigger spectrum. That's all of these disorders, okay? You're not alone, it's okay. We will come up with ways to better stratify, which means divide it into groups where we can understand the future better. We oftentimes, for natural history studies, know the later stages of disease, right? Because that's when children are getting diagnosed. They're oftentimes getting diagnosed years after they began to have differences. And so we can use natural history studies to look both forwards but also backwards through looking at old medical records and seeing, can we pull out from the records in a rigorous way ways to map the disease before we even knew it was there. And then another thing that we're working towards as a group is patient-centric outcomes. Talking to you all, meeting with you, having meetings, and coming up with, okay, what does a family think is important? What would you define as a meaningful improvement? And designing our clinical trials around that too. And now I'm going to pivot a little bit into where I think we can go in the community, and I'm going to use some experiences in other rare diseases. So bear with me. I think there are maybe some lessons that we can pull from it. I joined the MLD community, which is metachromatic leukodystrophy, a couple years ago. So in 2019, I was asked to join an FDA listening session. This is something I see in our future as a 4-H community, where I went as the disease expert with a group of families, and the FDA just listened to the families, and I gave context. And it was a beautiful and eye-opening experience for me as I really began to understand the importance of patient-centric outcomes for clinical trials. We then had a patient-facing drug development meeting, which is really the level up of that FDA listening session, where it was a full-day event where the families shared their stories in a formal, now open platform. And then for the first time ever, the FDA invited us back to actually have a scientific response meeting. So all the key opinion leaders joined together, and we helped to translate the patient perspective into how we think it could be made into a good clinical trial. And so it was really a wonderful and eye-opening experience. And from that externally led patient-facing drug development meeting, or ELF PFDD, we identified concepts of interest. Families over and over said that the ability to walk was important. Okay, well then how do we turn that into what we can measure? And so those are our outcome measures. And so you can start to map together what's important to families through things like the ELFPD into outcome measures for clinical trials. And one point on why do I care about clinical trials and natural history studies? Again, we are late in diagnosing most rare diseases. Outside of newborn screening, we don't listen to families, right? It takes years, sometimes decades to get a diagnosis. And when do I want to intervene at some point in the future? It's at the very beginning, right? And so we can actually take old medical records to help define how did a disease begin? What were the very first things families were bringing up to their physicians? And listening to those in retrospect and apologizing that they weren't listened to the first time. But now we can use those medical records and start listening. And if you take a rare disorder, and this is, I think, an example of MLD, the open circles are pre-diagnosis, and these are medical encounters. These families, and this is true for 4-H as well, are going into the medical system many, many, many times with concerns before the child is actually diagnosed. And so there are ways you can rigorously use this information as a platform to start mapping even before you knew what the diagnosis was. As a surprise to no one, when you compare when a child presents to when they are diagnosed, it's years, right? And that's really important, informative information to design clinical trials. I have been working with Anjana Sevagamurthy with our team and Sylvia Matua, who is a clinical research coordinator, to really come up with rigorous platforms for mapping this retrospective information. We take multi-site enrollment, meaning lots of groups can feed into it. We create unique study identifiers. We collect records across every single institution. So all the pediatrician visits, every single time you went to your neurologist, every time you went to your physical therapist, to see if there is rigorous information we can pull from that to not have to reinvent the wheel, to get as much information that we already have sitting at our fingertips. We then have a rigorous process where we go through, we extract the data through standardized protocols, and we can use this information to start mapping the history of the disease earlier. One of the things we can do too is, you guys, many of you have done outcome assessments through natural history studies. Well, a lot of that information, we don't have to have the patient in front of us. We can just look at the old medical records. And so we can start to score the old medical records to say, okay, the physician described the child as having some wobbliness with their gait. Okay, what score would I assign that? It seems a little subjective. So we actually have two physicians blinded to each other's score that look at the same medical record and try and assign the same score. And then we have a third rater come in and evaluate and see if they agree with the three. So it's through this process that you have to build in extra rigor, because otherwise it could be somewhat subjective or somewhat biased with what you know is going to happen. And I want to build in the final moments of my talk the story that we've done for multiple sulfatase deficiency. So to give you context, this is an even rarer disease than 4-H. When we began this work, we thought around 30 to 40 families in the world existed, so less than are sitting in this room right now. Because of my work in metachromatic leukodystrophy, I was invited to the first International MSD conference in 2017 by my friend Becca Ahrens. She was like, hey, do you want to go to Dublin with me? And I went, okay. And so very similar to where I hadn't seen a child with MSD before, but I was willing to jump into the space because friends said, I need your help. And I said, okay. And so I went to this first conference, and I met some really wonderful people there. I met Lars Skotala, who was an MD PhD at University of Göttingen. I met Steve Gray and Rachel Bailey, who developed gene therapy. And all of us just kind of got invited to this meeting. And the person that organized it is Alan Finglas. Where is he? He just won an award from the World Lysosomal Meeting, and it's going to be delivered this February as the best patient advocate in the world. he's awesome. And so I also met Brenna Bentley over the time and Michael Gelb, who develops newborn screening platforms, and Marzia Pascali, who runs ARUP, which is one of the big screening labs in the US. And it's through these friendships and networks of people that really just cared about an ultra, ultra rare disease, we began to start describing it. So this is a child that was born with ichthyosis or scaly skin, had some very subtle differences to their face. So you can imagine in 4-H, it's the teeth formation that's a little bit different, some bony abnormalities, but the kid keeps on getting told it's okay until ultimately they were found to have loss of walking, right? And it's when things started to change that people start listening to the families. And on MRI, there's, in this case, diffuse demyelination. So the myelin forms normally and then is lost. But you can see the parallels with 4-H. And 4-H is complicated. MSD, I think, is even more complicated. It's an activator enzyme. And so our bodies have 18 different cellular sulfatases, and every single one of them has to be activated, and you lose the ability to activate every single one of them, right? And so it is multiple sulfatase deficiency because these children are losing every single one of those sulfatases to some degree. And so it's actually a combination of many different monogenic or single gene disorders. And we were really historically only identifying children because they'd had regression or loss of skills. We were sometimes finding siblings who were affected presymptomatically. But the really exciting thing is because it is affects all sulfatases, this includes 2 sulfatases that are either on or soon to be on newborn screening. So we're going to be starting to find these children with this ultra, ultra rare disorder. So when we began, there was a need very similar to the 4-H community where we had absolutely no biomarker. We had no idea how to prognosticate or predict the future. We didn't know if we could reach the target that we needed to reach. We had really no natural history study, and we had no validated outcomes. And one by one, we made a list at a meeting just like this. We identified the needs, and as a team, we went through, and over the next 6 years, we hit every single one of these. And now we have 2 trials, right? We're going to have a small molecule drug reappropriation trial in Europe using retinoids this spring, and this work was also awarded this Bespoke Gene Therapy Consortium grant, and so we're going to have a gene therapy within the next 2 years in clinical trials for this disease. And it was because of a meeting like this and a family organization that was incredibly engaged that in 6 years we went from minimal knowledge, 10 families, to now 40 families are participating in our natural history study. Like, it is a family group that drove this. And so it feels overwhelming, the amount of work we have to do, but we know what we need to do, right? We just make a checklist and we go through it, okay? We were able to use our retrospective records to start mapping out natural history and find really clear phenotypes of the kids we thought changed over the period of time that would be good for a therapy, right? For a clinical trial, right? And so we identified which outcomes to use in these clinical trials using our retrospective records. We've now confirmed it using our prospective natural history study. We have started to classify or stratify disease into severe versus attenuated, right? So again, this challenge of which bucket. We've started to define the systemic involvement in these diseases and map it out and understand when and how it's affecting children across their organs. And then we've also worked to develop biomarkers for this disease. And we were lucky in some ways because children's sulfatases are affected and there are known sulfatase biomarkers. And so we had a hypothesis that because these children look like they really have metachromatic leukodystrophy, not multiple sulfatase deficiency, maybe their MLD enzymes are most affected. And maybe the MPS disorders, which are another type of sulfatase disorders that cause changes in neurodevelopment, those are affected strongly too. And so we thought maybe there would be distinct patterns that would help us at birth identify children that would either be strongly affected or more mildly affected. And yes, we ended up being lucky in that our hypothesis was true, and now we can identify a child before they have problems and start to stratify and better understand the disease very early. And so we've gone from a place where We knew very little about MSD. It had been in case reports only, to now we are in a place where we have a good genotype-phenotype correlation. We have newborn screening because we're going to piggyback on other disorders. And we have 2 clinical trials that are coming in the next 1 to 2 years. That's a really incredible place to be. But this is true across the leukodystrophies. I use the one example I am most involved in, but many of the leukodystrophies were a place with the same barriers, and we're starting to overcome them together. And so again, a moment of hope. We will be there, right? We're going to build the tools we need, the resources we need, because we have a strong and engaged community. This is our network across the studies that I have spoken about today. But the 4-H community is just as big and just as robust, and so we will be there in the future as well.